Bacteria whisper to our genes tiny molecules slip into nuclear receptors like secret agents. They mimic hormones we make, but the source is a colony, not a cell. How does this bacterial chatter reshape biology? These quorum-sensing molecules, called N-acyl homoserine lactones, carry a hydrophobic tail and a lactone ring. The tail slides into the ligand-binding pocket of receptors like PPAR-gamma, while the ring forms hydrogen bonds with key amino acids. This snug fit nudges the receptor's shape, exposing sites for co-activators. Once activated, the receptor binds DNA response elements and turns on genes that modulate inflammation and metabolism. In cell culture experiments, adding AHLs increases polarized fluorescence, confirming binding, and surface plasmon resonance shows micromolar affinity. These data demonstrate that bacterial signals can directly influence human gene expression through nuclear receptors. 0.8 nuclear receptors are transcription factors that sense small molecules. They bind ligands in a specialized pocket called the ligand-binding domain. When activated, the receptor changes shape to recruit co-activators. Co-activators then remodel chromatin and enable RNA polymerase access. The DNA response element is a specific sequence where the receptor dimer binds. Peroxisome proliferator activated receptors, or PPAR, respond to fatty acids and icosanoids. Liver X receptors, LXR, are activated by oxysterols derived from cholesterol. The aryl hydrocarbon receptor, AR, senses xenobiotic compounds like dioxins. Each family shares a conserved DNA binding domain but differs in ligand specificity. Ligand binding promotes heterodimerization with retinoid X receptors, RXR. The heterodimer then attaches to the response element and initiates transcription. Thus, quorum-sensing molecules that mimic these ligands can hijack host gene regulation. 0.8 quorum-sensing molecules, or QSM, are small bacterial signals that travel between cells. All, a common class of QSMs, have an acyl side chain and a lactone ring. When an AHL enters a host cell, it can bind to nuclear receptors. The ligand-binding pocket of these receptors is designed for endogenous fatty acids. An AHL fits into this cavity, forming hydrogen bonds with key amino acids. Simultaneously, hydrophobic contacts stabilize the complex and shift receptor conformation. This structural change alters how the receptor interacts with DNA response elements. Consequently, transcription of target genes is either upregulated or downregulated. Molecular docking simulations illustrate these interactions by overlaying an AHL inside PPAR-gamma. The visualization highlights hydrogen bonds and hydrophobic contacts that stabilize the ligand-bound state. 0.8's fluorescence polarization measures how a fluorescently labeled AHL tumbles in solution. When an AHL binds to a nuclear receptor, its rotation slows. The resulting increase in polarized light indicates complex formation. By varying AHL concentration, we plot polarization versus ligand amount. This curve reveals the dissociation constant, Kd, for the interaction. Surface plasmon resonance offers a complementary real-time method. AHLs flow over a sensor chip with immobilized receptor protein. Binding changes the refractive index, producing a measurable signal. The sensorgram shows association and dissociation phases, from which Kd is derived. Both assays confirm that AHLs bind nuclear receptors with micromolar affinity. These quantitative data support the hypothesis of bacterial host signaling crosstalk. Thus, in vitro binding assays provide mechanistic evidence for quorum-sensing ligands. 0.8's bacterial quorum-sensing molecules, such as acyl homoserine lactones, diffuse across the mucosal surface. Epithelial cells capture these signals through membrane transporters, or direct diffusion. Inside the cell, AHLs bind to specific host nuclear receptors like PXR or R. Ligand binding triggers receptor dimerization and exposure of a nuclear localization signal. The complex then moves into the nucleus within minutes. Once inside, it attaches to DNA response elements and promoter regions of target genes. This attachment initiates transcription of cytokine genes, such as IL-8 and tight junction proteins like claudin-1. Resulting protein production strengthens barrier integrity and modulates local immune tone. Simultaneously, the receptor activates metabolic enzymes that alter bile acid composition. Thus, quorum sensing molecules orchestrate a coordinated host response to microbial presence. Pause 0.8's quorum sensing molecules can bind host nuclear receptors, altering gene expression. This ligand-receptor interaction provides a mechanistic bridge between microbes and host cells. By mapping these interactions, researchers identify specific microbial signals that influence metabolic pathways. Engineered probiotic consortia can be designed to produce or sequester such signals. In a conceptual diagram, each bacterial strain is linked to a receptor target. Arrows in the illustration represent sequential research milestones from discovery to validation. Early studies focus on characterizing binding affinities and downstream transcriptional changes. Subsequent work evaluates how altering signal levels shifts host physiological readouts. These insights guide rational design of microbial communities that modulate receptor activity. Future directions include high-throughput screening of natural QSM libraries for receptor specificity. Integrating omics data will refine predictive models of microbiome host crosstalk. Ultimately, this knowledge supports the development of microbiome-based strategies with defined mechanistic outcomes. Pause 0.8's quorum sensing molecules can fit into host nuclear receptors, altering receptor shape and gene expression. Experiments show micromolar affinity for PPAR-gamma, leading to anti-inflammatory gene activation. This crosstalk may influence immune responses and metabolic pathways.